Okay, hi. So I'm very happy today to introduce Julia Hockenmaier. Many of you probably know Julia's work on CCG, combinatorial categorical grammar. So she was one of the first people to use this in a large-scale or statistical way. And she has a whole series of papers culminating in her dissertation at Edinburgh, where her advisor was Mark Steedman. And she's actually created CCG versions of the Penn tree bank and of a German tree bank and so forth, which have both been released by the LDC. Is that right?
The German version is not the least, but yeah. But
the English version is.
The English one is.
And we can get the German one from you if we need it.
Eventually, yes, once it's done. Yeah.
Julia was also here in the, I think the 2003 workshop, 2002 workshop, working on the team doing code training for parsing, for bootstrapping parsers. And she's also done some psycholinguistics work related to using CCG and priming. But what she's talking about today is what she's been doing for the last few years as a postdoc at Penn, working with Aravind Joshi, and also working with Ken Dill's lab at UCSF on using methods from parsing in computational biology, and specifically to model the process of protein folding. So this is really interesting cross-disciplinary work, and we're looking forward to hearing it.
Thanks, Jason. How long is it supposed to be, like 15 minutes?
Yeah.
Okay. I'm going to try, yeah. The problem with coming sort of on the train and having slides on the laptop is that you sort of end up adding more and more stuff on the laptop, so I'm going to try and be quick. Okay, so, well, I used to think that sort of these two facts don't have anything to do with each other, right? So we know that people understand language, by and large, and we know that proteins, which are these biomolecules that are in all of our cells, that they form into these unique three-dimensional structures. Actually, do we need to use a microphone, or is that okay?
We hear you.
You sound fine.
Okay, cool. So, yeah, nothing in common, right? Totally separate problems. Except that, of course, they're both really, really hard for computers. And I guess I don't need to tell you much about why language is important, right? You all know this, CLSP. But you might not know why we actually care about protein folding, or why we want to be able to predict these structures. And, well, the motivation really comes from big pharma, right? It's really, the main reason is probably because you want to design new drugs. So a drug is something which binds to a protein, or which is a protein, and you want to make sure that it binds to the right protein and not to the wrong ones, and so on. So in order to do that, you have to be able to predict quickly the right structure for proteins, and for many proteins. So people think that computational techniques, if they work, are going to be much quicker than anything you can do in the wet lab. And the other reason is also medical, and that's that there are a lot of diseases that are due to protein misfolding. Alzheimer's, CJD, so, mad cow disease, etc., etc. There's just lots and lots of things that happen to go wrong in your body if your proteins don't fold correctly. So there the idea is that you really have to understand the folding process. It's not good enough to just be able to predict the structure. You really want to understand what goes on. So what are proteins? Well, so they're sequences of amino acids. So you can just think of them as sort of these beads along a chain, and the chain is called the backbone. And there's 20 different kinds of amino acids. And they all differ sort of in these side chains, which are sort of abbreviated here just as R's.
Excuse me, I'm completely naive.
Yeah.
Amino acids you pointed to.
Amino acids
are these things.
These things.
And what are the proteins? The whole thing?
And the protein is a whole chain of amino acids.
I see.
Right. So it's what's called a polypeptide sequence.
And is there a finite alphabet of amino acids?
There's 20 of them.
There are only 20.
There are only 20
so
of them.
The proteins are just the combination.
Exactly.
Right.
The interesting thing is that not all random sequences over this alphabet are actually foldable proteins. It's sort of like in language, like a random sequence of English words isn't going to form a sentence, right? In 99.9% of the cases. And the same for proteins. Most random amino acid sequences aren't going to fold into anything.
And fold means that it parses.
Fold.
So to speak.
We're going to come to that. Yes,
Yeah, we
yes,
will.
yes, yes. That's yeah.
So, sorry, quick question. I mean, I hate to have so many interruptions early on, but when you say doesn't fold into anything,
Yeah.
I can't imagine what that would mean.
Okay. So it means it doesn't have a unique structure it folds into.
Thank you. Okay. Yeah, okay, it's
it
not
means
stable.
It's not stable, or it doesn't have. Yeah.
But those chains are observed, even though they might not fold into.
Or you can
No, the
synthesize.
thing is that your body produces fold into something, and if they don't, you have a problem.
Okay.
If you don't, yeah, you might not live that much longer. it's, yeah. So even though there are 20 different amino acids, there's sort of two main classes that we need to worry about, and so there are the hydrophobic ones. That means they don't want to be surrounded by water.
You don't want to be sucked.
They don't want to be surrounded by water.
Surrounded by-
Right, okay. And then the other ones, polar or, yeah, there's other ones. So that's going to be really important because the protein, your cell consists actually mainly of water. So these hydrophobic side chains aren't going to be very happy in the cell, right? They don't want to be surrounded by water. So energetically, it's favorable for them to be sort of buried. Right? And that's actually what drives the whole folding process. It's called the hydrophobic effect. So folding means that you have some sort of random chain, some open chain conformation, and it's going to fold spontaneously into one unique structure. And in this unique structure, parts of the chain that are really far apart in this open chain conformation, so for instance this part and this part here, they end up being really close together spatially. And these are side chain contacts, right? So these contacts between these amino acids, they can also be used to characterize, for instance, the topology of the folded structure. I'm going to be talking a lot about the native state, and all I mean by that is the lowest energy structure, the folded structure. In this native state, all of these hydrophobic side chains end up on the inside of the molecule. So they all end up being sheltered from water, from the solution, and that is, as I said, the main driving force behind folding.
You need a 3D animation.
Hmm?
I think you need some 3D animation to show us.
Yes, I should have a 3D animation. Yes, there are plenty of those on the web actually. I know, yeah. So one thing we know, well, one of the few things we know for a fact about protein folding is that so the topology of these dependencies that determines how fast it's going to fold. So if you have something like a helix, that's sort of like, so it's sort of like a screwdriver. You have lots of local contacts, which I've shown you here. So if this is the chain, then in the helix you're going to have lots of local contacts. That's going to be a really fast folding protein. Whereas if you have something, what's called a beta sheet, so that's sort of this kind of a structure. We have lots and lots of contacts here. That's going to be really slow to fold. And you're going to come back later to that.
And slow is good or bad?
Fast is much better.
Fast is better.
Yeah. Although it doesn't really matter, right? I mean, well, for the computer, it's going to be easier to model fast-folding proteins because it takes a lot of time to model slow-folding proteins. Right? Also, one thing that so if you come from like the parsing community, you might think, well, this looks sort of just like a dependency graph in some sense, and you could think of them that way, just as sort of as these dependencies or contacts between these amino acids. And protein folding research over the last 40 years has been driven by this one question. So when you think, so this guy Cyrus Leventhal, who in the 1960s did this simple back of the envelope calculation. He said, well, the average protein has got 150 amino acids. How many possible structures does this have? Well, it's around the order of 10 to the 300. So it's totally impossible for it to search everything. But it has to be able to find the right structure. Otherwise we wouldn't be here, otherwise we wouldn't live. How is this possible? How can the protein find its right structure? So protein folding is a really, really interesting search problem. And of course it's
Excuse me, are we to think of it that to start with, this thing lies around like this, and then it starts folding, then it's actually a dynamic process?
Yes. So the way people have found this out is that, well, it's difficult to observe in the cell, right? But what you can do is, with most proteins, you can take them just in a test tube, and then you can, for instance, change the temperature or change the pH value, and then the thing will unfold. But then if you change it back again to into folding conditions, it's going to fold spontaneously into its right structure. So that means that so that's why people think that the structure is the optimal structure thermodynamically and so on, because this process happens spontaneously, and that's also why we know that all the information has to be in the sequence, because under the right conditions, there's nothing else that could influence this process. It just has to be sort of the energetics.
But in a living cell, when the protein is first synthesized,
Yeah.
is there a period of time where it exists in its unfolded state? Or does it fold?
That was
Because
my question.
Because it's built left to right, if I'm not
Yes.
wrong, or whatever, in some
Yeah,
order.
yeah.
So does it just fold
So-
as it's growing, or does it first become a little string dangling around, and then it scratches out?
I've read papers that said that it's sort of that it's sheltered first, sort of, so as it gets, sort of synthesized, it gets sheltered until the whole thing is synthesized, and then it gets released.
So some sort of
But-
sheet would sort of stick to it and make sure it doesn't have any tendency to collapse in itself. And when it's all done, it would suddenly disintegrate and the thing would start floating.
Yeah, I've heard that sort of what's the case, but I'm not sure. It could be. I mean, so the physical or the biophysical folding problem has always been posed as this question of, in the absence of anything, this thing has to fold. How does this work? That's sort of the way the problem has been posed. I'm not sure what exactly the theories are about what happens in the cell. I think there could be arguments about sort of,
Because I could imagine that if you're thinking that the hydrophobic elements are somehow coming together and closing in on each other, then that while they're being synthesized, there's clearly something next to them. And if that
Yeah.
could prevent them from being attracted to each other, that protein could stay unfolded as long as those molecules are there. And when they go away, that's when it stops.
Yeah, exactly. Something like that could happen. Okay, so now we're faced with sort of two unrelated search problems, right? So natural language parsing, which
So one question, can the protein fold into more than one structure based on different conditions? If you have a different condition, it will fold into some other structure. You have
there are
some-
these sort of moonlighting proteins that I think do something like that, but the proteins that people typically study don't. There's always one unique structure. So in parsing, as we all know, we want to find the right grammatical structure of a sentence. Right? Whereas in protein folding or protein structure prediction, we want to find the folded structure of a protein chain. So these are really, really actually similar search problems, right? In both cases, we have an input sequence. And in both cases, there's a hidden structure. And we need to know the structure in order to make use of that sequence. And there's a lot of ambiguity, so we have to be able to find the optimal structure of that sequence.
What do you mean hidden structure? It folds itself to whatever it folds itself, and then it's not hidden, or is it hidden?
If we just have the it's not, once it's folded, it's not hidden. But for instance, if you just look at the genome, right? So you know that a particular gene encodes for a particular amino acid sequence. Like, if
You're just saying you want a model that predicts the gold standard, right?
Hmm?
You want a model that predicts the gold standard before the gold standard is revealed.
Yeah, exactly.
Well, the
exactly.
difference is that here the gold standard actually exists. It's not in somebody's mind. Whereas
Well,
in
actually, it's all
parsing-
throughout the body, including that.
I don't get it. I don't understand this gold standard. Where did gold standard come from?
The gold standard is the actual physical structure.
Where does a gene come into this? We
Okay.
knew we had amino acids and their
Okay,
sequence
genes-
created proteins. Where is gene now?
Okay, gene, okay. So DNA, right, it's an alphabet of 4 letters.
Yes.
Right? And like every sort of, every triplet codes for one amino acid.
Every triplet of those 4 letters.
Yeah.
Okay.
Yeah, because of one amino
Yes,
acid,
yes,
and
okay.
that's so genes encode amino acid sequences, right? And then these amino acid sequences, they fold in, they're proteins. They actually have specific functions in our cells, like the enzymes or, so let me.
Well, I still
So-
don't know what the gene is. Gene is the DNA.
Yeah.
The whole DNA is one gene.
No.
No,
No.
the DNA is a long strand. Some strings of it are called genes because they actually code for a protein.
For a particular protein.
Yeah. So, right.
A gene codes for one protein.
So out of these 6
Oh,
billion
gene codes
base
for
pairs
one
that-
protein.
Yes. Each gene codes for one protein.
It's mainly what
Alright.
genes do.
All right.
And so the amino acid sequences has a 1-to-1 mapping with a sequence of base pairs.
All right.
Please.
Okay. Right.
Yeah. So we have a lot of, for instance, genomic data, but we don't know. But unless we know what these proteins fold into, we don't know what
We don't know.
we
which
don't know
is-
how to use the because we don't know exactly what the function.
So there is a question of segmentation. In other words,
Yeah,
there's a
yeah.
string of amino acids and certain ones.
Yes, but I'm not
certain
going to deal with
sequen-
this. I'm not going to deal with this today. So in protein folding, you just assume you know the protein, but you would anybody put a good structure.
I see. Okay.
So in other words,
So
I
it's
could
a
ask the
very-
abstract question. I could give you a strand of DNA, which is some sequence of ATGCs.
Yes.
And I could say, if this were a gene and
Yes.
it synthesized a protein, what structure would that protein fall
Exactly,
into? So I
exactly.
would give you the ATGC sequence from which you would construct the amino acid sequence.
Exactly.
And then you'd ask, given a chain, a backbone with these things hanging off of it, how will
Exactly.
it fold?
Exactly.
Okay.
Yeah. Okay. So and in both cases, so in language and proteins, the structure is determined by the sequence. Yeah.
I have a question regarding this analogy that we are using.
Yeah.
In protein folding, actually the structure is determined not only by the constituents of the sequence themselves, but also by the external conditions. Whereas in sentence structures, it would only depend on the constituents.
Yeah, I'm just assuming under folding conditions. I'm assuming under folding conditions. Right? Under folding conditions, you have a unique native state. Right.
Let me move that we let Julia get through some more slides.
Yeah. Okay. Yeah. Okay. And so how do we solve these search problems computationally? Well, we need a way to represent all possible structures, and we need a way to score these structures, right? So in language, that would be a grammar and a probability model. If we're dealing with proteins, we need a way to represent protein structures, and the scoring function would typically be called an energy function. It actually turns out to be equivalent to a probability model, but it's the same thing. And we also need a search algorithm. Okay. So today, I'm going to assume a fixed structural representation and a fixed scoring function. I'm going to explain those to you in a minute. And I'm just going to focus on the search algorithm. And in particular, I'm going to be using a language-based search algorithm, or a search algorithm that has been developed to analyze language. to address the folding problem. Was there a question in the back? No?
We haven't had enough slides yet, so. No.
Okay, sure. Okay, no questions. Okay, so the question is, can we use parsing algorithms for protein folding? And then the big question in protein folding is always, does this algorithm allow us to avoid exhaustive search? And then the second question is, can it help us understand how real proteins fold? And okay, the only problem is that real proteins are really, really difficult to simulate, right? So either you need a petaflop supercomputer, or you need a project like Folding at Home at Stanford, where they sort of ask thousands and thousands of people to donate their screensaver time. Okay. I don't have these resources. I'm just a postdoc. I might have access to a cluster, but neither of these resources. And also they haven't really had, they're still dealing with really short proteins, right? Only 21 amino acids or 36 amino acids. Not really the best way to explore novel search strategies. We have to go simpler. And I'm going to go the simplest.
That's right. What they're doing is some kind of backtracking search, branch and bound, or something like that.
They use sort of molecular dynamics kind of simulation, so they really model every single atom.
I see. Got it.
And they write down sort of Newton's equations and so on, and they have these force fields, and then they just simulate the whole chain all at once.
Right.
Quickly.
And what you're trying to do is get similar results. You want to know where every link in the background is and at what angle.
Yeah.
Exactly. That's going to be the output of both models, but they're doing it by direct simulation.
Exactly. And so, except that I am not going to be using real proteins for now. I will in the long term, but in my current work, I'm not quite there yet. I'm going to be using the HP model, which is one of the most simplest sort of models of proteins. It's just
HP stands for Hewlett Packard.
H stands for hydrophobic and P stands for polar. So it's not a printer. It's not if you search for HP model, that's typically what you get. You get some printers. So folding is still an NP-hard problem in this model, even though it's the simplest possible model. So what we do is we assume that space is discretized. And in this particular case, we're going to assume that space is only two-dimensional, and it's like a chessboard. So you have these squares or these lattice sides, and we're going to assume that the protein is a chain. Well, as it is, right? The protein is a chain, and that each amino acid, each bead, can be on one of these lattice sides. And we're going to maintain chain connectivity by assuming that beads that are adjacent in the chain have to be on adjacent sides. And we can either go left or right or up or down, but we can't go diagonally. And also, once a square is occupied, you can't put another square on it. We have two kinds of amino acids, hydrophobic and polar ones. So the H are, the black ones are the hydrophobic ones, and the white ones are the ones we don't really care about. And in order to model this hydrophobic effect that I told you about, we're going to have an energy function. And this energy function is what's called the contact potential. So you count how many of these black beads you get close to each other. Obviously black beads that are not adjacent along the backbones, right? So this won't count because that's the same for all possible structures. So we just want to get structures that have the highest number of these H-H contacts. And we're going to deal with a subset of sequences that have a unique lowest energy structure. People have enumerated those up to like 25, so we know that. So that's the gold standard in the system. It's exact enumeration. People have known it, right? And again, this is just because it's a toy system where we want to explore the properties and so on.
Before we go on,
Sure.
So why do you, is there any reason to count how many P's are on the outside of the structure?
Yeah, people look at that. People look at sort of at the surface ratio and so on.
Right.
But there are papers on that. Yeah, but the energy function, hang on, now I've got to go through this. So the energy function typically doesn't take that into account. I mean, you could imagine some other term that takes it into account, but we don't.
Okay. And then the other question is, why 2D? Why not 3D?
Hmm?
Why 2 dimensions?
There are, because it's,
I mean, you could do the same in 3, right?
people also use a three-dimensional variant of this. We have a cubic lattice, yeah. I'm just going to use the 2D variant here, but you also have three-dimensional variants of it. In 3D, the combinatorics sort of get really bad. You have to assume that you're just in a really compact cube or something, and yeah. But it's okay. So now let's think back about this folding as a search problem.
And one last question.
Okay.
In the energy function, is there any penalty for leaving a hole in it?
No. There is. So you mean like?
You know, I could.
Like a big
You
loop.
showed me
Like a loop.
a chain with exactly 16 amino acid
Yes.
things on it and a 4x4 square. But in real space, the space is unlimited, and I can
Yes.
make a little circle out of it or some other structure
Yes.
that has a hole in it.
Yes, you.
There's an opportunity cost to doing
Yeah, you don't. Yeah, exactly. You don't end up with structures like that. You end up.
No, there
It
may
is
be an
really-
opportunity
yeah.
cost. My question is there a penalty for it?
Not an explicit one. No. No. But you end up with very complex structures that don't have loops. Because if you have a loop, in your lowest energy structure, you typically don't have a unique folding sequence.
Well, I mean, you haven't convinced me that this will have a unique solution yet. But
Only a small percentage of sequences does. And we're only going to deal with those that have. Okay, so let's just think again about this folding a search problem, right? So the question was, how can the protein find its one structure among the 10 to the 300 possible solutions? Well, so in some sense, this question, well, I mean, this only arises if you think that the energy landscape, well, so an energy landscape is a mapping from all possible structures that the protein has to their energies, right? And you can sort of think as folding as a random walk along this landscape. And so if you think that the energy landscape is sort of like a golf course, where like all structures have got the same energy except for the right one, then you're always going to sort of wander around aimlessly, and you're never going to fold, right? And it's going to take you forever. But we don't really think that that's what's going on. So the original solution by Leventhal was to assume that there's a single pathway, so that so it's sort of like that. So you start in one part of the landscape, and you're always going to reach the final state. We don't really think anymore that that's what's going on, but that's sort of what has motivated people to look for folding pathways or folding routes. That sort of. What people really think is that the energy landscape looks more like this. So it's like a funnel, and you might have sort of bumps in there and traps, and you can sort of start anywhere along the funnel, and you can go downhill pretty fast.
Excuse me, all three cases are illustrating the same folding? Is the folding the eventual thing? so to
These
speak?
three things are all different folding pathways. You can sort of imagine the protein starts in this configuration here, it's so open chain configuration, and it goes downhill until it finds the folded state.
And the folded state is the same?
And the folded state is the same down here in the bottom, right?
And that's all that counts?
Yeah, exactly. You just want to reach that state down here. Yeah.
and these are conjectures about the true energy landscape.
Yes.
What about the model you just gave us?
Yeah. The model also has an energy landscape that sort of look. You can draw it in different ways, right? But it also has an energy landscape. Well, actually, that's the next slide. Okay. So hang on. So one way to think about these structures is to say I'm going to group them, and I'm going to group equivalent structures into the same group, right? And because the energy function only cares about these H-H contacts, I'm going to group all structures with the same H-H contacts into the same class. So for instance, here are all the structures. So this is just a sixmer, and
Six what?
a sixmer, like a chain of 6 beads. And here are all the structures without any contacts, except I think one of them does have a contact. This one here, that shouldn't be there. But that have to do things by hand. Here are all the structures with one contact or with another contact. And here, so for this particular sequence, you don't have a unique folded state, but you have 3 different sequences with 2 contacts each, right? So one thing you see is that the more contacts you have, the fewer corresponding structures you have, right? Here you have a lot of structures. Here you just have a few, and here you only have 1 for each set of contacts. And the number of structures is called the entropy, or rather the log of the number of structures called entropy. So you can think of the energy landscape as this. So this is how you end up with this sort of funnel shape, because there's a lot of high energy structures and only very few low energy structures. So how does folding work? Well, one theory is the sort of zipping model, where folding starts by the chain growing, local structures in different parts of the chain at the same time. And then at some point these different structures assemble and come together. So for instance, so you have like a model of hierarchical folding. So you have sort of these local structures, and then at some point they come together, they assemble. Well, when you assume this, then, and when you think again then about the folding roots, and you believe this hierarchical folding, then the folding root is actually a tree over the sequence, right? So I'm just going to show you the same thing again as before, oops, and you can see it's a tree.
So each step in that was a neighbor within the topology of the disk that you were showing before.
Yes.
When you were talking about folding roots, that was all very abstract. You weren't saying which states of the protein were adjacent to one another.
Yeah, exactly.
But now you're saying that 2 states are adjacent if you can get from one to another by doing that on some part of the protein.
Exactly. That's a local search. And here is a figure from a paper from 1979 by someone who's actually faculty here at Hopkins. And what he did is he went through all the known structures at the time and showed that you could actually you recursively decompose them into these domains. And if you do that, you end up with a tree structure. We also know that when you watch a protein fold, which is difficult to do, but you see that some fragments fold faster than the rest of the chain. We also know that for some proteins, if you chop them up and you just take a small fragment, that fragment is going to fold by itself. That's not true for all fragments, but it's true for some fragments.
Can you just clarify that sentence again? Proteins have recursive domains. It means that a protein has what? It has parts, and each part has its own folding scheme, and it will do that the same way no matter what the other part is?
Just structurally.
Okay.
Domains refer to the folded structure.
So the folded structure.
Yeah, so you can,
So an example a counterexample would be a spiral in your model. If you had a spiral, it would be, maybe you could still get the spiral by, it would just be a right branching tree or something.
Yeah, exactly.
Okay.
Or any balance tree.
Yeah.
Or any.
But yeah.
Yeah. So is there any way to falsify this statement that protein? I mean, let's stick to your 2D model just for simplicity.
Yes. There's evidence against hierarchical folding, and that is that there are some proteins where people say, well, okay, when we watch this fold, we actually see that the ends come together first.
I see.
And that can't happen in a tree. And we're going to come back to that later.
That's only dynamic evidence, though. So if you look at the final folded structure, that by itself can't give evidence against hierarchical folding, right? You could always come up with a tree post hoc that could have produced it. Is that right?
Yeah, I mean, I'm going to make the assumption that all the partial structures that correspond to a node in a folding tree are local minimum, right? Sort of energetically lowest energy structures for this particular fragment.
Okay. Yes, and then there may be no way to do it.
And
Right.
yeah, exactly.
Yeah. Okay. Thanks. That's helpful.
Yeah. but this experimental evidence here, for instance, the evidence against hierarchical folding, that comes from an actual experiment, an actual wet lab experiment, and this theory about hierarchical folding that really it's a theory, and also, well, we're going to think of it as a computational model, right? And in most cases, when you have a theory, or when you try to come up with a mechanism for protein folding, you want to explain how individual molecules behave, right? How does this molecule go from this structure to that structure, and so on, right? But that's not what experiments observe, right? Experiments observe a whole ensemble of molecules, right? You have a whole test tube full of proteins, and they might all be doing different things at the same time. So you have sort of something like this, where your theory is expressed in terms of the behavior of one individual molecule, and your experiment actually sees a whole bunch of them. And again, we're going to come back to that later. So the question is, how are we going to implement this hierarchical search? And well, being computational linguists, we're going to try CKY chart parsing. And I presume you all know what chart parsing is, but I'm just going to go through.
No, there are biologists in the audience.
Oh, right. Okay, good. Okay.
So now it's our turn to laugh at them, since they've been laughing at us for the first time.
Right. Okay. Okay. So here's a sentence: we eat sushi, and here is something that's called a context-free grammar. So we know that eat is a verb, and we and sushi are noun phrases, and that we could in English we can form a sentence if we have a noun to the subject that's followed by verb and something, and the verb and something is called a verb phrase, which consists, for instance, of a verb and its object. Right? So, for instance, we can eat something. So we can use this grammar to analyze the sentence. And so what we do is we first create what's called a parse chart. So that's a table where we first start with the words, and then we have the pairs of words, and then we have the whole sentence up here, because we just had 3 words. So, okay, let's fill this chart. We start, we is a noun phrase, goes in here. Eat is a verb, goes into the cell. Sushi is a noun phrase. And then, well, there's no rule that combines NP and V in this grammar. But, oops, sorry. Sorry. That's the problem with animations. So, okay, we eat, there's nothing we can do. But we can put eat and sushi together, right? Because we have this one rule here. So we have V and NP.
Why can't you put we eat together? Because it's not there.
Because there's no rule in this particular grammar.
What would the rule have to look like?
You could have a unary rule.
In this case, it would be a rule that rewrites to the right of the arrow. You'd have to have NPV.
NPV, yeah.
We and eat together.
Can you explain all these rules? What does that mean? S arrow NPV? What is that?
It means that you can, well, so the way these rules are written is you say.
Do you want to draw a tree on the blackboard? That might help.
Sure. Yes. So this actually corresponds to a tree that looks like S, NP, VP, and then.
Just draw the 3 rules separately.
Okay.
No, not
So
one.
you imagine you start off with a single symbol S,
So S-
and what those rules say is how you can rewrite those symbols. And you iteratively rewrite that string of symbols until you come to a string of words.
Or a better way to think of it is that the things on the right, if they happen to be next to each other, you can assemble them to create the thing on the left.
Yeah, which is what this algorithm does. Right? It starts at the bottom and it sort of works its way up.
So actually, if you made your arrows point the other way, it would make.
Yes, it would, because that's what the algorithm does. Yeah. Okay. Anyway, now if I move to the next slide. Oh yeah, and then also we can extract these trees, which so this tree here is the same tree that I just drew here, except turned around. And so now let's look at doing this for the HP model. So what we're going to do is, because our energy function only cares about the hydrophobic interactions, we're going to split the chain into small fragments such that each fragment only contains one of these black beads. And in fact, we split them in the middle, not the way I've shown you here, but it took too long to draw these figures, so I'm not going to redraw them again. And then we take these small fragments, and then we just enumerate all their possible structures, and we put them into the chart. And then basically we just play jigsaw. We just put these adjacent pieces together in any possible way.
When you have these structures, there is a marked left endpoint and the
Yes,
right end.
exactly.
And only the right endpoint of one can go with the.
Exactly. Yeah. Yes. You want to maintain chain connectivity in the right way, of course. Yeah.
And was it a problem that it's not unique how to cut these things? Was it unique how to cut these things?
No, you can define it in any way, really. You don't want to have more than one H.
Yeah.
Well, but you don't want to have any initial H contacts.
Right.
Because you don't want to commit to those. Anything that goes into these initial cells is something you commit to. So you don't want, so you don't want to say, my full state has to have this particular contact.
And it seems that it won't make a difference how you cut it anyway, because any dangling P chain that you left on this side, had you left it on that side,
Exactly.
you could have assembled it straight
Exactly.
the way this one is assembled.
It turns out that it's a bit more efficient, on the average, if you have more or less equally sized fragments, and you
When you
will
say
have
fragments, you mean tails.
You mean these? I just mean these little bits of the chain, right?
For equally sized chains rather than equally
No,
sized.
fragments. Right.
Okay.
Right.
I'm sorry, there's something key here that I'm not getting. How can you guarantee that you'll find the optimal solution if you didn't break things up amino acid by amino acid?
Oh, okay. Well, first of all, the white bits, they don't contribute to the energy.
Sorry?
The white beads, they don't contribute to the energy, so it doesn't matter.
So you can push them around as you feel like.
Yeah, exactly.
You can
They only matter.
Okay,
Like,
okay. You're cutting them at every dark bead.
Yeah, exactly.
Yeah,
exactly.
okay, now I get it.
Exactly.
Okay, there's only one dark bead per die.
Exactly.
Yeah, okay, thanks.
Yeah. But yes, there is no guarantee. There is no guarantee for this algorithm to work.
Because you're assuming hierarchical fold.
Because, there is no guarantee that this algorithm is going to work because in every cell I'm only going to keep the lowest energy structures, and there's no guarantee that these are going to
You're doing pruning.
I'm doing pruning,
Okay,
exactly.
thanks.
If I
You're doing
didn't do
unsafe
pruning, if I didn't do any pruning at all, this would be completely crazy. This would be completely insane because I would be enumerating the whole thing like sort of n cubic time.
Sure. So basically, you're doing these equivalence classes by. What you should be, the non-terminal should be the complete description of the boundary of the structure so far, because that's what that differentiates how it interacts.
Yeah.
From other things with different boundaries.
Yeah, exactly.
But you're only keeping one best per cell. So this is like
Well,
parsing.
I'm
where
not
you,
keeping one best per cell. I'm keeping all of the lowest energy structures. I don't.
Yeah, one best with ties, but
Yeah.
the-
Exactly. Yeah.
Sure, but if you have, this is like having noun phrases
I
and
thought
verb
we
phrases
had a
repeated.
moratorium on questions.
Well, that was
That
when
was
we
a
all
long
had
time
time
ago.
slots. Go ahead, sorry.
Okay. There was just one. Yeah.
We account for degeneracy somehow?
Yes. Yeah. I
It
do.
was the chart of yours?
I take care of symmetries. Right. I only have one equivalent. Yeah.
No, you had the chart where you showed all the equivalent things.
Oh yes, yeah. I
Exactly.
do. So there are a lot of sort of mirror symmetry and so on the lattice, and I take care of those. And otherwise, you can group these structures by their contact maps if you want to. And we're going to use that later, actually, in the second half of the talk, which I hope we're going to get to.
We've had a lot of questions because of the dual audience, so if you need a little extra time, that's okay.
Okay. So I'm going to be quick. So basically, you're going to do exactly the same thing, right?
How did these things disappear?
They're not
pruning.
lowest energy.
I mean,
Pruning.
I see.
Yeah.
So each of these guys.
With an evaluation function.
With the
Inside
energy
class.
function. I use the energy function.
Use the inside class, right?
Yes. I don't have an estimate of the outside cost. I could. It would be good to do that.
Yeah.
But yeah. So and then hopefully at the end, the top cell contains the folded structure, and then we can extract these folding trees. And actually, here's a paper again by the same people from 1981, but they looked at real proteins and they hypothesized their folding routes. And if you're a computational linguist, you can, well, so this is the sequence here. And I think these are sort of contacts. Then, I mean, this is really just like a packed chart in a way.
This is a parse forest.
Yeah, it's a parse forest. Yeah. This is in a, this is in a protein folding paper from 1981. I really thought it was cute. I was very happy when I found this. Okay, well, so what we get in our model is, so here I've shown you a 25mer, or 24mer, I think, which sort of has this hairpin and it comes right here, and then you have a helix, on the side of it. And I'm just showing you one particular parse tree. And here I'm showing you the chart. And what the colors indicate is where all of the folding roots that lead to the native state lie. So black means that all of the roots go through it. So all of these initial fragments are of course contained in the tree. And the top cell here is also contained because we have fold structure. And then you have sort of different ways of putting things together, which is why you have different shades of gray. But basically that's sort of more or less one folding pathway, more or less, right? And which sort of corresponds to this tree. You really have to grow the structure in one particular way. So there's a lot of misfolding here, right? Anything that the algorithm searches here is sort of isn't quite right. Is not going to lead to the native state. And we end up with a lot of these patterns, right? I mean, typically sequences have these specific patterns. And just to show you how well it works, well, so we looked at all the 20mers because that was still a sequence length we could deal with exhaustively. So it's about 25,000 sequences. The way you can arrange a 20mer on a lattice, corresponds to about 42 million different ways. And if you just count the number of sort of different things that CKY tries, it corresponds to about 2.5% of this number. And CKY finds the native state for 96.7% of these structures. So there's no guarantee, but it works pretty well and it's really fast. There are faster ways of predicting the right structure. You can use, there are some heuristic ways that work much, much faster. But what you get here is all of these folding roots at the same time. That's something you can't get from any other algorithm. And if you know about CKY and binary trees, you know that CKY is the most efficient way of searching for binary trees over an input sequence. So
it's not actually all possible folding roots because of the pruning, right?
Yeah, under the assumption that, yeah, exactly. I mean, you can play around with the pruning criteria, actually. That's something
Yes.
you can play. Okay. One thing I just want to show you real quick is that the chart actually defines an energy landscape. So here again, I've shown you a sequence. So again, from the first to the last monomer, and these x's are contacts. So this is something that biologists call a contact map, and it looks just like a parse chart, right?
Tell me again, what are the axes?
So the axes are just, I have a contact between this H and that H.
So that means there is a phrase from the H to the H.
You could think of it that way, yeah. So these two things come together in the folded state, yeah.
And
And then
these are all the possible contacts that could be had in this string. That's what you mean.
Yeah, I think so. I don't think that all of them are native, but I don't remember exactly. But
I don't see the tree now.
No, there's no tree yet.
There's no tree. I'm just showing you all the possible structures you can find for this particular sequence, which is sort of what CKY does. And then of course you can think that each cell in your parse chart has got an energy level, right? Depending on the lowest energy you find, right? And you're going to start with an energy of 0, and then for each contact that you find, you're going to go down 1 energy.
I don't understand this contact business. I'm sorry.
So this means you can find a contact from
the H will contact H. It's folded so that the two H's are going to be next to each other.
Yes. Yeah.
In the final tree.
No, just whatever you can find. Whatever you can, and I'm showing you whatever you can find in this particular sequence.
Let me see if I can interpret this. Every large rectangular square here is a hypothesized constituent, and the reason that those aren't individual cells is because you've already pre-segmented your input. And the color of a rectangle is given by the energy of, or if you prefer, the Viterbi inside cost.
Yeah. So so each of these sort of, well, I'm showing so the contact map is usually given in terms of the actual amino acids, right? Which is what these fine lines show you. What the thick lines show you is the fragments that the algorithm splits the chain into. So these correspond to the cells of the parse chart.
Right. The thick outline boxes.
Yeah, exactly.
Every thick outline box is a cell of the parse chart.
Exactly, and I've just projected that back onto the actual sequence.
Yes, exactly.
Right.
And the color is giving you the score of that cell.
Exactly. And of course you go down in energy as you...
And the X is there if, in virtue of doing that folding, you would get contact between two H's.
Exactly. So once you do that, and of course you can turn this around, and then you end up with an energy landscape. Right? And what's interesting is that these chart landscapes, they actually determine the amount of search that CKY does. So it's really, really fast if you can prune early, right? So here's the main diagonal, and this means you have some local contacts. And so if you have a really steep funnel, then you're not going to require much search. But here you have sort of these big plateaus, right? And so the number of structures is exponential in the length of the sequence. So actually, once you're up here, you have a lot of structures to carry around with you, because you can't prune anything, right? And if you're in a situation like that, if you're dealing with a sequence like that, your algorithm is going to take forever. And again, this just sort of comes back to this sort of the funnel landscape versus this sort of Leventhal paradox sort of golf course landscape. And another thing we can do that I'm not going to have time to explain is that we can calculate these computational folding rates that actually predict really well this contact order correlation that I sort of mentioned briefly at the beginning. But it's really nice because it shows us that there really is something to this. So hierarchical search, it works pretty well. I mean, there's no guarantee, but for the sequence we looked at, it seems to work. And
when you say work, meaning it folds that way?
Yeah. Well,
if you find
it predicts the right structure, yeah. And because we can sort of, well,
predicts like structure, that means the structure which ends up in that upper corner and has lowest energy is most of the time
correct.
Could be always.
Exactly.
Right?
Yeah. Exactly.
Is the only thing you're testing really the pruning algorithm? Because if you didn't prune, it would always work, right?
Yes. You are testing the pruning algorithm, and once you move to a different energy function, you would have to play around with the pruning function.
If you change the pruning algorithm, does that affect the correlations on the previous slide with the actual empirical folding rates?
I haven't tested that yet.
Could you go back to the previous slide?
Let me hypothesize that you're not testing the pruning algorithm. What you're testing is your assumption that the actual folding happens through a sequence of minimal energy subfolding. Because it's a dynamic program, there is no pruning at this point. And it's just a dynamic program saying you keep the highest, lowest energy in every cell.
Right.
And if there are multiple, you keep all of them. So in that sense, there is no pruning going on.
In that
And what
sense,
you're testing is the hypothesis that
that works.
the true minimum energy configuration is attained through a sequence
through a local
of local
search,
minimum energy.
Exactly. Yeah.
And in 98% of the cases, that is true.
Yeah. Exactly.
I guess the question is in the remaining 2%, how far away is the minimum
By what?
energy attainable by local optimal folds?
What I've
compared
seen
to
only by
global
one, but you're only missing one contact.
Okay. And are these the cases where the ends have to come together first?
Not necessarily. Yeah, I don't know. I haven't really looked at them so many.
Could you imagine that a little local search using some additional kinds of moves would be able to fix up those remaining 2% of cases?
Possibly. I mean, one thing we know is that the energy landscape of the HP mold is a lot trappier than that for real proteins. So I'm not, at this
I
point,
see.
I'm, you know,
Sure, this is all artificial.
I don't really want to spend that much more time on a lattice model. You know, long term I want to move away from it. I move to real proteins. But so this is really just a proof of concept. And I do actually have a second half of the talk that I'm going to try, and it's a really, really quick. So because I think it's actually perhaps more fun than the first part. So okay. So of course we know that proteins don't really use dynamic programming, right? They might form a contact. It might be the right one. It might be the wrong one. So then they unfold, and then they defold, and so on, right? So they go through this whole cycle until eventually they find the right state.
And we know this because somebody has observed it with a microscope or something?
Well,
and how do we know that it does it in this?
Well, we know that there's misfolding. There's no reason to assume that the protein knows immediately.
No, but somebody has observed that a protein that has started misfolding badly has corrected itself and then folded correctly.
Well, yeah, we know for some proteins, for instance, there are some proteins that that are beta sheets. Sort of like the. Yeah. And we know that sometimes we see these folding intermediates, which are helices. Right. So clearly, there's misfolding happening.
So they've been caught red-handed in unoptimal forms.
Yes, but do they correct themselves?
Yes, they do. They do, because they do end up in the right structure eventually.
I see.
Right. Okay. So what do we do? Well, okay. And also, what we want, what we really want to do is we want to predict the collective behavior of a whole ensemble of molecules. Right? And well, okay. So assuming that folding is hierarchical, we want to build a Markov chain model of this whole process, and we're going to use dynamic programming to construct this chain. For each protein, we're going to have one specific Markov chain that models the whole folding process. Okay, so I presume that most of you here know what a Markov chain is. It's just, well, if you're a computer scientist, you can think of it as a weighted finite state automaton. What really matters is that you have a set of states and you have a probability distribution over these states, and you can move between these states in certain prescribed ways. And the Markov chain gives you a time-dependent probability distribution over these states. So if you have some initial distribution, you know the probability distribution at any time, sort of t. So we need a set of states. We need a transition rate matrix which defines how you can move from one state to the next state and slices between these states. I want to assume continuous time, not discrete time, but it's very similar to what playing. To the discrete time case that might be more familiar to those of you who work in language. And once we know the set of states and the transition matrix, and we assume there's a initial distribution, we know what the probability at time t is going to be. What are our states going to be? Well, we're going to assume that folding is hierarchical and that direct folding routes are going to be trees. So in this case, a state is just a tree cut. So it's a horizontal slice through the tree.
State is what?
A state is a tree cut. It's a horizontal slice through the tree. Right? You want to capture the whole protein. Right? So that's always going to be some horizontal slice through this tree. Right? You have to span the whole chain. And, right. So you end up with these horizontal slices. And we're going to label the nodes with contact maps or equivalence classes. One thing that's kind of interesting is that now we don't really have binary trees anymore. We can actually have flatter trees. So if you think about this chain here, 1, 2, 3, 4, you can, and if 3 is also likely, you can have 2 equivalent binary trees, but in effect, it's just one ternary tree. And we're going to define these transition in terms of these folding trees. So we're going to assume that the way you can move, so you can move up or down the tree in one part of the tree. There's just one local transition between 2 children and their parents, which is a folding transition, or the other way around, which is an unfolding transition. So here's just a quick example. Here's a chain. You can have the contacts 1, 4, 5, 8, and 1 and 8. You can't have 1 and 5 or 4 and 8 because of the geometry of the square lattice. So in hierarchical folding, we can go from the unfolded chain to 1, 4, or to 5, 8, and from both of these we can go to 1, 4, and 5, 8. And each of these we can go to this to the native state which has all three contacts. But there's a number of things we can't do. For instance, we can't form this contact first, 1-8.
Which contact? I don't see where.
1-8. We can't form 1-8 directly. There's no, in hierarchical folding you don't have that state. It's just an example that's built into the model. Again, that's this case where the ends would come together before the inside is folded. Okay. And the way we're going to calculate these transition rates, well, physically they depend on the difference in free energy. We're just given by this formula here. So you need to take into account the difference in energy, which is the difference in the number of H contacts. Then you have sort of this temperature constant, and you have the difference in entropy. The entropy, again, is log of the number of conformations. Well, we know the energy exactly, and the entropy is an approximation. And we use these local transitions to calculate these rates. And I can show you some exact enumeration results that show that even though it's an approximation, we end up on average with the right result. Which is a good thing. There are a few little tricks we have to pull with, but basically we end up with the right thing. And then there's a specific way of getting from this delta G value to these transition rates. And how do we actually obtain this Markov chain with dynamic programming? Well, what we're going to do is we're going to construct this weighted finite state automaton in each cell of the chart. We're going to start in these initial cells with really small, minimal finite state automata for these little fragments, and we're going to end up with one big graph in the top cell, which is going to give us the graph for the whole cell. And the reason why this is going to work is that, so the states are contact maps. Right? So in each initial cell, right, each of these initial fragments can't have any contacts. So we just have one state, and the state is going to be the empty contact map. Right? And then when we combine two cells, the way we combine these graphs is basically by forming the cross product. But in all cases, you're going to end up with a single source in this graph, which has no contacts, right? So that's why you end up with one unique graph in the top cell. I'm just going to run you real quick through this example again. So we're going to split the chain. We're going to end up with this chart. And
why did you split it the way you split it, and not 1 and then 3 zeros?
Because it's more efficient. Just enumeration, it's more efficient if you split it in equal bits.
Okay.
Okay, so for 1, 2, there's no contact, 3, 4 is no contact, and so on. 1, 4, again, we have no contact because so that's the cross product. But then we're also going to find the contact 1, 4 in this cell. So that's going to be another node. 3, 6, there's no contact possible, so just the empty state. 5, 8, again, just one no contact or 5, 8. 1, 6, nothing. 1, 4, and nothing. 3, 8, nothing. 5, 8. And then the top cell, I'm going to show you here, we have nothing. We have 1, 4, we have 5, 8, and 1, 4, 5, 8. And 1, 4, 5, 8, and 1, 8. Which is the actual node up there, which is the lowest energy structure. Yeah, here's just some pseudo code. So basically what this algorithm allows us to do, if you take no word for it, because I didn't really have time to explain it, is it allows us to capture this whole hierarchical folding process, starting from the sequence alone. Because now we can construct this Markov chain just from the sequence alone.
So you're saying you don't have to take random walks?
You don't have to sample, sort of the algorithm does it for you, right? It searches the whole space. Right? So again, we're going to look at this particular sequence here. And the Markov chain we end up with has 193 states, which is pretty small. And okay, what I'm going to show you is how this protein folds. What I'm going to see. So here's this time on the log scale, and the y-axis shows all these 193 states, and they're sorted by the energy. So at the top you're going to see the high energy states, so the unfolded state, and at the bottom you're going to see the actual folded native state. And the colors, I'm going to show you the log probabilities of these states. So okay, so red means high probability, blue means low probability. So you really see this thing sort of falling down the funnel. And also what you see is that sort of some streams are quicker to reach than others, and which is why you end up with a sort of banded structure here. And at the end, everything is folded.
Can you get a second order graph that tells you how often you're fixing misfolds?
You could. I haven't done that yet. There's lots of things you can do. I mean, you can look at a population of misfolded chains over time and so on.
In this case, I was just asking about the distribution over transitions, like how many transitions are improving energy and how many transitions are holding energy. So you can't read that off this graph. It's a second order graph.
I don't know how to get that out of a continuous time chain. I don't. There must be a way. Yeah.
That's a good question. I think I can answer it all.
Oh right. Okay. Yeah. Well, of course, what experiments see isn't really log probabilities. You're not. If you look at real probabilities, you're going to see everything unfolded, which is this tiny red band up here, and then at the end, everything folded. What experiments might actually see, so again, now this is real probabilities, is sort of substructures. In particular, you're going to have, for instance, here you have a helix. Okay, so these are not log probabilities, these are actual probabilities. And this is the same time scale. So you can see this helix being formed relatively early on. And you can see this hairpin here. And that's formed much later. And you can actually see that this hairpin, once you've found this hairpin, you found the whole structure. Another thing you can look at is the probability of these end-to-end contacts. So just 1 and 12 or 2 and 13. Now that's really strange. Okay, so this is time on a log scale. What you see is that this happens much earlier than this hairpin. How is this possible? Right? There's no folding route that allows us to do that. Why? This is really bizarre. Is there some sort of a bug in the system, or? Well, it turns out no. This is really what you would observe, and the reason why you would observe this is because, well, for instance, you can also look at this contact 2-5. Well, that's formed relatively early on. But there's another contact, 3-6, and it turns out that is actually sort of higher populated early on. And the reason for that is that I mean, it's a bit hard to see just from the structure here, but actually there's a lot more combinations with 3-6 than with 2-5. So 3-6 is a lot more likely than 2-5. Well, and if you have 2-6, and 3-6 actually leads to a trap state, which is identical to the later state, except for this contact 3-6. So when you see, and you can see that, I mean, that's never very highly populated, but it's populated, well, it's populated enough that when you just look at the probabilities of these end-to-end contacts,
native means that this occurs naturally? Is that
native, yeah, that's what it means in real proteins. Here it just means it's the lowest energy structure.
Okay. And why isn't 16 connected to 3 in the native?
Oh, it is.
So why don't you have the number then?
That would have been a yellow line. In other words, the chain itself doesn't have a connection. The chain goes 1, 2, 3, 4.
Here I just
the chain ends at 16.
So maybe connection is, but is the point that 2 is with 5 and 1 is with 6?
Yeah.
So
I'm
why
just looking. So if you were to just look at 112 and 213, right? And if you were to ignore 313, right? Yes. You would end up with this sort of observation that the ends, sort of the ends, like meaning 1, 2, and 12, 13, they come together early. And the reason, why you might think that, right, is because of this trap state, which happens to be, to have exactly those contacts as well. Right. So that really means what? So actually, there's evidence that the ends come together early. It's not clear what that really means. means, right? Because it's not right? I mean, there's no single folding route that allows this to happen, and it's not really clear that these macroscopic observations, right, which you get by looking at a whole test tube, right? So the posterior probability of anything really tells you something about the microscopic moves in this model. Yeah. So, and of course, if you think about probability, if you think about it in terms of probabilities, then it kind of makes sense. But I think it's important to bear in mind, just like.
So I think in the experiments, those are 5-value analysis. Is that?
Yes.
So those are usually whether it's before or after the transition state.
Yes.
Do you know where the transition state is in time there?
I haven't really looked into the transition state yet, but you can do phi analysis. So okay, so phi analysis is basically just you change one amino acid into something else, and you look what happens to the folding rate and so on, and where it gets stuck. So
I guess my question comes from that in that graph. It looks like those seem to be folded before the final state, but they still look pretty late on your plot. So is it?
That's because it's a log scale. Time is not a log scale.
So would that be equivalent to what would show up in the 5-value analysis? I guess it would kind of depend where that transition state is.
It would, yeah. I haven't really looked into doing phi analysis yet, but you could, yeah, definitely. There are some papers by Manu Oskin who did that for similar kinds of Markov chain models for lattice proteins. So yeah, but yeah. Ultimately, of course, I want to go beyond the lattice, and and of course, the details need to be worked out. But basically, I'm pretty optimistic that it can be done, because dynamic programming is, of course, a general tool, and CKY is easily parallelizable, right? So each of these combinations sort of can be found off to a different node on the cluster and so on. And you can even use sort of contact maps and contact energies to talk about these structures, although you might want to use a more fine-grained representation. And the people that I work with in San Francisco, they've used similar kinds of hierarchical folding ideas, although not really in the same kind of systematic way in the protein folding competition that ran last summer, and it worked really well. So just to conclude, yeah, if you just focus on the search algorithm part of the folding problem, then if you choose the right kind of search algorithm, which I think is dynamic programming for this case, it can give us new insight into the folding process that might be difficult to get otherwise. And that's really it. So thanks.
Well, it seems like nobody has any questions.
If you do, please, yeah.
It's supposed to be a joke.
Well, does anybody have any questions, or otherwise just email me or?
People are welcome to stay and ask questions. I really was just joking.
I was wondering how you can, hypothetically implement atomic resolutions version of this algorithm.
Well, one thing you could do, so what the Dill lab did in CASP is they sort of implemented these contacts as springs, right? So you can sort of say, I'm going to do, so each of these local combinations is a small electrodynamic simulation. And each step, I'm going to find some local energy structures, and I'm going to fix these contacts as springs. And then I'm going to rerun my simulation, sort of putting these two fragments together. You really have to, obviously you can't play this sort of jigsaw game anymore once you're in continuous space, right? That really only makes sense in discrete space. But yeah.
In reality, the residues are not either for both, or they have characteristics that can relate.
Yeah, of course. But you can still think of sidechain contacts, right? You have to think about how exactly am I going to define them? Am I going to sort of just take some cutoff, or right? People do define contacts for real proteins, right? a lot of details that still have to be worked out. I'm sure there's a lot of things to be done.
So I'd like to understand better the final algorithm that you gave, where you
yes, of course,
dealt with all the paths of the continuous time Markov chain, but we can talk about that after the talk.
Yeah, sure, we can just take it off.
Are there any other questions before we thank the speaker again? Jeff, you?
Just out of curiosity, I mean, the protein stuff is really fun. How does this help you in linguistics to sort this out? Is that a really brief, I mean, because it's, what exactly are you searching for when you make one of these trees?
Well, so you don't really have physics to give you an energy function, right? But you have sort of something that's like the PDB. So you have these tree banks where people just sat down and like annotated 1 million words of the Wall Street Journal with what they think is the right structure. So you have to have some consensus as to what is the structure of that.
Definition of what the
Right,
structure
yeah.
is.
You have a
There's
linguistic
a lot
energy
of
function.
contention there. There are a lot of wars about how to represent structures. Right? We all sort of agree more or less on what a sentence means, but how to represent structures is, that's what linguistics is about, to a large extent. But you do want to know these structures, and then you can use these data sets to estimate probability models. Right? So I mean, just like you do statistical potentials for protein folding, it's sort of the same kind of an exercise. And then you can play these competitions just like you do with protein folding. Yeah. It's sort of, yeah.
Good. Well, thanks very much, Julian, for tolerating all of our naive questions. And let's thank the speaker again.
Thanks a lot for asking me.